Welcome back everyone to the final installment of Unit 1 DNA in the Genome, Genomics and Genomic Sequencing. Genomics, of course, determining the entire sequence of bases in an organism's genome. Bioinformatics, a relatively new field in biology, combining molecular biology, statistical analysis, along with computer technology. Now, genome sequencing and genomics can be used for the study of evolutionary biology, inheritance, and personalized medicine. If we firstly have a look at genomic sequencing, firstly it involves enzymes called restriction endonucleases, and these identify within the DNA strands restriction sites. These are very short sequences of nucleotides, about 4 to 8 nucleotides long, and they will cut the DNA strand at that point. And of course, just like any other enzyme, restriction endonucleases are specific to a particular restriction site. So firstly, in what's called the genome shotgun approach, many copies of the original DNA strand that you're trying to sequence are made using PCR, and the sequence in each fragment is determined, and they're cut with various endonucleases so that these fragments can ultimately be arranged in correct order once each fragment has been sequenced. Like so, you have fragment P, fragment Q, and fragment R. Those three fragments, there would be considerable overlap between them. Now, the actual process of sequencing the DNA, of course, the DNA template, the original strand that needs to be sequenced, is copied many times by PCR, and that requires what you would expect: DNA polymerase, primers, and a supply of 4 nucleotides, of course adenine, thymine, guanine, and cytosine, along with the DNA template, of course. But in genomic sequencing, there's the addition of what's called modified nucleotides, and these have been treated with a fluorescent dye. Now, they act completely normally when they're being incorporated into a growing complementary strand. However, what happens is, once they are incorporated, the polymerase will stop replicating the complementary strand at that point. Okay? So here we are in the process. There's our copy of DNA strand to be sequenced, or our template strand. The short sequence at the bottom there matches with the primer. And here's the region, for example, here's the region which we are trying to determine in terms of the sequence of bases. We have our supply of normal nucleotides, we have a lot of primers, and we also have our supply of modified nucleotides. So that reaction mixture is allowed, all mixed together, allowed to proceed. And what we should have, as long as there are enough template strands of DNA, and as long as there are enough primers and normal nucleotides and modified nucleotides, then we should have one of these modified nucleotides appearing at every single base point along the template strand that we are trying to determine, okay, at each base position along that template strand. Okay. What happens then, once that reaction has been allowed to proceed, you take that reaction mixture and you apply electrophoresis to it, and that's basically separation using an electric current. So the smaller molecules will travel further than larger molecules, and what you ultimately have there is your sequence of bases as they appear on the complementary strand. Now, of course, if that's the sequence of bases on the complementary strand, then the actual template strand you are trying to determine is like so. And all that can be done using an automated sequence analyzer. Now, the fragments are sequenced by the analyzer. Now, because of the way the DNA strand's been cut, the fragments, there will be lots of overlap, okay, and the actual sequence of fragments in which order they should be placed is the actual sequence of that is helped by the specific endonucleases that cut the fragments at various points, and the computer will then work out which order the fragments should all be in relative to each other, and hence the entire genome of the organism you're trying to determine. If we move on to genomics, now the entire human genome mapped using that shotgun method and samples from just a few donors, but other organisms' genomes have been determined, for example pathogenic viruses and bacteria, such as the bacterium that causes tuberculosis, various pest species like mosquito, and the malarial Plasmodium protozoan, and of course model organisms that possess genes similar to ours, like in the common mouse. We can use genomics to compare the genomes of different organisms of different species to determine the similarities in their genomes. Members of the same species, such as harmless E. coli and E. coli, which could cause disease, and the differences in cancerous and normal cells to see how different mutations cause cancerous growth. Now, differences in genome could be due to what are called single nucleotide polymorphisms. These are essentially point mutations, and they can be revealed, and their importance revealed by bioinformatics. And in this case, there's the single substitution point mutation there. Can also be used to determine similarities in genomes. There are many sequences in the genome which are called conserved sequences, such as the active sites of essential enzymes. And another example, the male sex determining gene of whales and humans is only different by 4 nucleotide pairs. Moving on to phylogenetics, this is looking at the evolutionary history and relatedness between different species by comparing the genomes along with the fossil evidence. And of course, more related species will have more similar genomes, and these can be used to construct phylogenetic trees tracing an organism's evolutionary history. Now, as part of the separation of related species of organisms over time is this concept of divergence. That over time, different groups of closely related organisms will acquire their own set of mutations, and over time they will form and diverge into genetically distinct groups. So the basic rule of thumb being that the more different the genomes of these species or groups of species is, then the more distantly related they are to each other. So the use of phylogenetics brings up this concept of evolutionary distance, meaning the number of differences in the base sequence per unit length of DNA. The greater number of differences, the greater the time since those different groups diverged away from each other. And as such, it's been determined that certain molecules within organisms can act as what's called molecular clocks, that over time, various nucleotide substitutions will appear in the genomes for these molecules, and the differences in them will be proportionate to the length of time since these different groups diverged from each other. For example, alpha-globin molecules, they're used in oxygen transport in many animals, in many organisms, and of course the differences in the genomes or the genetic sequences for these globins will give an indication of the time since divergence for various groups. Now, if we compare molecular phylogenetics versus the normal observation of animals in terms of their structure, that organisms such as sharks and tuna, even though they look very similar, they are very much more distantly related than dolphins and bats are, even though their morphology, their body plan, is essentially far more different than between that of a shark and a tuna fish. Now, the only downside to the use of molecular dating really is that it assumes that mutation rates have remained constant over time. And of course, that being the case, that if you are trying to compare the genomes of very, very distantly related organisms, then molecular dating is less reliable because of that. Now, another use of molecular phylogenetics has been to determine the degree of relatedness between the 3 main domains of living things, and they are domain bacteria, your traditional prokaryotes, the archaea, which are mostly prokaryotes, but they include organisms that are termed as extremophiles, which live in extreme environments like volcanic hot springs, such as our old friend Thermus aquaticus from PCR, and the eukaryotes, as we know of as fungi, plants, and animals, including ourselves. Now, from this, we can see that the archaea, even though they're very similar in form to bacteria, they do possess some similarities with eukaryotic cells. Okay, and it's also found that mitochondrial and chloroplast DNA originated in prokaryotes, and these organelles were adopted by early eukaryotic life forms. So, genomics and fossil evidence can be used to determine the major sequences of evolutionary events on the planet. And if we look at the picture on the left-hand side of the screen, you see that all up to this point, only unicellular life existed on the planet Earth. But most of this was in the sea at this point, of course. So if we determine the sequence of major events afterwards, animals and vertebrates would have come earlier in the sequence of life on Earth than land plants, as life evolved in the sea first of all, and it was only when land plants began to evolve that animals could then progress onto the land. So another use of genomic studies and genomic sequencing is personal genomics, and this is becoming faster and cheaper these days as the genomic sequencing procedures are becoming more readily available. They can help determine genetic disorders. In reality, it's very difficult to distinguish whether mutations are harmful and which are neutral. Genetics disorders are caused by variations in the genome, and a link will be found between the mutant variant in the genome and the genetic disorders or diseases. So when a mutant variant in the genome causes a genetic disease or disorder, it has been known as molecularly characterized. But genetic disorders are caused by a combination of genetic and environmental factors, of course. But so far, about 2,200 genetic disorders have been discovered through genomic sequencing. This could help in personalized medicine. Various people may react differently to different drugs, and to reduce the possibility of side effects, personal genomics could be used to determine which medicines are the best for individual patients. Of course, it could also be used for determining risk prediction to see if you're going to be susceptible to any of these disorders and others, and to determine whether people are going to be predisposed to certain genetic disorders or diseases in the future. However, there are issues related to this. Who records that information, who holds that information, and who has access to that information relating to individuals, such as would insurance companies perhaps wish to see any personal genomics information before you're able to take out various policies. Okay, thank you very much.